Welcome to the cBioPortal tutorial series brought to you by the Genomic Education team at the Jackson Laboratory. This video will demonstrate how to locate individual patient data within a study on the cBioPortal. Using the cBioPortal, it is possible to locate data for individual patients within a study. Here we are within a pancreatic cancer study, and we are on the landing page, which is within the Summary tab of the study. In order to locate individual patient data, we must navigate to the Clinical Data tab. By clicking on this tab, we navigate to a list of patients and samples within the study. The patients have been given a patient ID so that they remain anonymous. In this study, the patient IDs begin with the letters PDA, which is then followed by a number. Also in this study, one sample was taken from each patient for genetic testing, so the sample ID and the patient ID match, as you can see here. On this Clinical Data tab, the data is displayed in this large table. The data will vary by study, but often includes relevant clinical demographic data such as sex, race category, and diagnosis age, as well as genetic data such as mutation count. There is also information about the patient's tumor stage at the time of diagnosis, as well as about therapy. For this particular study, data on radiation therapy is included. To look more closely at a specific patient, you can click on the patient ID. By clicking this number, a new browser window will load with more detailed information. The landing page is the Summary tab, and on this tab, we find genetic information, including a list of gene variants uncovered through genetic testing on this patient's tumor. Let's first focus on this table in the middle of the page. This is a list of gene variants identified in this patient's tumor, or in other words, a list of mutated genes. This patient had 36 genes with mutations identified in their tumor. The gene names listed as their official symbols, which are a combination of letters and numbers as we see here, are in the column on the left under the name gene. The next column indicates the protein change. The genes included in this list are protein coding genes, so there will be a corresponding protein change that accompanies each mutation. Let's look at an example. For this top gene in the list, KRAS, we see that the protein change is listed as G12V. The glycine, that is normally the 12th amino acid in the protein sequence, has been changed to a valine for this variant. If we look at the column labeled mutation type, this correctly identifies this mutation as a missense mutation. This was likely a single base exchange in the gene, leading to a single amino acid substitution in the protein. The other types of mutations include deletions or insertions. The letters FS that precede DEL for deletion or INS for insertion indicate the presence of a frame shift in the coded protein as the result of the mutation. Another type of mutation is a nonsense mutation, which is when a stop codon replaces an amino acid in the protein sequence. This results in a protein that is shorter than normal due to a stop codon coming earlier in the sequence as the result of this mutation. By default, the genes in the table are listed in order of clinical significance, indicated by this bullseye symbol located under annotation. To learn more about clinical significance, please see the other tutorials in this series. It is possible, though, to sort the table by another column. If we were interested in identifying different types of mutations in this study, we could sort the list by clicking mutation type. We see now that there are 2 deletions with frame shifts and 1 insertion with a frame shift. There are also many missense mutations as we scroll through the table, And then finally, at the end, we see 2 nonsense mutations. It is also possible to search for a gene in this list if you are interested in looking for a specific gene. Say we are studying a gene called MCM4. If we type this gene name, MCM4, into the search bar on the top right, we can see that this patient has a missense mutation in this gene. If a gene is not present when it is typed into this search bar, this will result in no genes found, such as if we type BRCA1. There are no results for this gene search. Also on this summary page, we can see 2 other sections: the copy number alterations, as well as a visualization of the genetic changes identified in this patient and their locations on specific chromosomes. Copy number alterations, shown towards the bottom of the page, are very common in cancer cells and represent DNA changes that are either insertions or deletions that are very large. I will not be going into detail about copy number alterations in this tutorial. But scrolling to the top of the page, we can see the chromosome visualization. We see the list of chromosomes in the human cell, which are numbered 1 through 22, and then the sex chromosomes X and Y listed here. And then we see 2 rows underneath. The first is the mutations, and the second is the copy number alterations. Let's look at an example. Here on chromosome 1, we can see that there's 1 mutation indicated by this green box, and several copy number alterations listed here below. If we click on number 1, we can see a more detailed diagram of the chromosome, which spans this distance here. And we also can see the annotated genes, which are indicated here with these blue boxes and diagrams below. If we click on the mutation, a box appears showing that there is a missense mutation in the gene RYR2. The location for this gene, or its chromosomal position, is provided here in this box as well. If we'd like to navigate to a different chromosome, you could use this drop-down menu, select another chromosome, and look at the mutations that appear on that chromosome. Mutations in coding genes can also be visualized in the Pathways tab. Many genes that are mutated in cancer cells code for proteins found in signaling pathways in the cell. The Pathways tab provides a visual of these pathways that are commonly studied in cancer cells. This patient has a missense mutation in the gene KRAS, which is very commonly mutated in cancer. If we select the RTK-RAS pathway by selecting that button there, we can see the gene KRAS, and if we hover over this gene, we see that there is a G12V missense mutation. If you look at the whole pathway, or you can scroll here, we can see that this pathway controls proliferation, cell survival, and translation. This is how KRAS mutations contribute to the progression of cancer, influencing these cellular processes. The final tab is the Clinical Data tab. Much of this data is included in the table that includes all of the patients, but here you can find additional demographic data as well as information about the tumor sample that was subject to genetic testing for this study. Here we see the type of cancer, the tumor stage, as well as mutation count. Thank you for your attention. To learn more about the cBioPortal and its features, continue to watch videos in this tutorial series. For other resources from Jack's Genomic Education, find us on our website.